name's Yatunde for B, and
my name's Dara Aarons.
And today we'll be talking to you about flow cytometry.
Oh.
So the history of flow cytometry is that it was developed in 1968 by Wolfgang Gaud, and that was the very first fluorescence-based cytometry, and that was also the same year that it was patented. So basically there are 6 components of the flow cytometer. There's the flow chamber, there's the laser, there's the light detector, the filter, the computer, and lastly the color detector. So I'm going to talk to you now about how the flow cytometry works. First of all, a petri dish or tube or vessel that contains the prepared sample that you want to analyze is put in a flow cytometer. Let's say this. And then the sample is drawn into a vessel and pumped into the flow chamber, which is this. And this happens one at a time. So there is a sheet of fluid that helps with this. It helps with friction. So it helps the cell go in and avoid friction about down here when it's squeezing into the flow chamber. So a laser beam hits the cell as they pass through the flow chamber, and the way the light bounces off the cell gives the information about its characteristics, which is about right around here. How the laser bounces. This is the laser, and those are the lights. There's the forward angle light scatter, which is this, and then there's the side angle light scatter, which is this. That's 90 degrees to the forward angle light scatter. So light scatter and fluorescence are captured, they're spectrally filtered, and they're directed into the appropriate photodetector for conversion into electrical signal. So the light scatter is collected in 2 different angles, as I said, the forward angle light scatter and then the perpendicular light scatter. So now I'm going to tell you what they do. Forward scatter measures scattered light in the direction of the laser path, as well as the size of the cell. Side scatter measures light 90 degrees to the laser path, which is the forward angle light scatter, and the granularity of the cell. So because the fluorescent light emitted by the cell can go in either way, forward or side. So as the cell passes through the light, fluorochromes are attached to them, and they're excited and they fluoresce. They emit a special color. So this red cells, like let's say red blood cells, or just any cells in general, this might exhibit this shade of light, this exhibit this shade of light, and this exhibit this shade of light. So they're then directed into the color detector. The color detector collects the different light that are emitted by the fluorochromes. Then the electron of the cytometer amplifies and processes the resulting data. It then converts analog to digital data that is stored on the computer, which is one of the 6 components of the flow cytometer. And then the data can now be analyzed.
Flow cytometry is used for a multitude of things, such as these listed, and in examples such as in marine biology, you can characterize the abundance and community structure of photosynthetic plankton by using autofluorescent properties, and in medicine for transplantation, hematology, tumor immunology, chemotherapy, prenatal diagnostics, genetics, sperm sorting, diagnosing blood cancers, and evaluating the progression of diseases such as AIDS. The cost of flow cytometry differs depending on the facility that it is being performed in, the cost of the machine, which can run upwards of $10,000, the cost of the reagents, and the researchers that are doing it. Each test can cost anywhere from about $3 to $22 per test. Some of the advantages are that you can use cells of many sizes, you can use a large library of antibodies, you can spot abnormalities in the cell, and you can simultaneously record multiple physical and chemical characteristics of the cell. Some of the disadvantages of flow cytometry include that there's a higher cost on some tests, must have viable cells for analysis, the cells must pass through the flow cytometer one at a time, which can be very slow, especially for some experience, and the whole machine must be calibrated, which can take a very long time.